And we're just starting the transit at 700 meters. Copy that.
Maybe a target middle. Hard to tell. Yeah, I think it's just particles lined up.
Mike, this is Brian on the X. Would you like to be running with the lasers on the Woolhark feed in case something large pops in, we can get a size on it?
Something big on sonar, 7.5. Middle left. They just scattered.
I'm not sure if I had mute on when I just said that. I don't think you'll like to be able to use the lasers on anything that you see in midwater.
Yeah, we can keep them on just in case. If that works, it doesn't really matter to us.
Large target coming middle. Looks like a fish. It's going off to the left side. Left side. It's moving slow. Left side. Come up just a touch. Right there.
Looks like we've got some kind of siphonophore or something coming into view right now. They're going to stop and try and get the ROV around it, take a closer look.
Okay, I just got my line 1 feedback, and that's good.
Excellent.
It just keeps jumping in and out. That's locked up again. Heck, I just lost the event log as well.
We can still hear you loud and clear on
That connection is
VoIP.
bad.
We still got you on the conference call.
Bridge RV now. What's our speed right now? Roger that, thank you.
All right, I'm back again.
Something maybe left side. I think it's gonna be too high. Come up there. Yeah, lost it. Three minutes remaining.
Everything appears to be working now.
Excellent. Yeah, hold you back there once you lost it. What I'll do is, if you call from Zoom, I'll just zoom in and out. Looks like a copepod or amphipod. The pilots were going to try and get a zoom, but he evaded us successfully.
Something center. Right there.
Another siphonophore coming into view.
Almost back to 58 degrees. See those footballs going there. Looking good. Getting a lot of feed here. Go, bottom right. Going.
See something just on the edge of visibility, probably a Tina 4 that we're going to try and get a zoom in on. Yep, looks like a Tina 4.
That's good. It's coming at you. Yeah, it's on the back of it.
That's a good shot.
Time here. Okay. He's got his streamer off the left.
Nice mid-water column macro.
And 700 meters.
And I've got good video also.
Excellent. We'll try and stop if we see anything, Mike, but yell anyways if you want to see something specifically.
We're going, yeah, 1340.
Okay. There's a fish. It's probably a cyclothony.
Got two objects coming in, one kind of gelatinous in nature, and jellyfish, and then another, can't tell if it's a ctenophore, next, just out of view to the right also.
Yeah, the one toward, just to the right of the center is a mucus web from a larvacean. The other's a jellyfish.
Ah,
The mucus webs are really important in carbon transport into the deep sea.
zooming in on something now, I'm not quite sure what it is.
Now, it's just like more mucus.
Yeah.
They're getting pretty good at zooming on that stuff in midwater, though.
Yep, that they are. Looks like another mucus net.
See, something else. It's another mucus net. A very nice zoom.
Pilots and video. Compliments are raining down from shore. Great job. Looks like another mucus net zoom. It may be my imagination, but I feel like we're seeing fewer copepod darting around in the screen as the depth has increased.
Comes in on the lower left. That seems to be streaming back to him. North one right next to the left base? Yep. That is squishy.
We're zooming in on something again. I'm not sure what it is. Possibly another siphonophore.
Yeah, nice zoom on the siphonophore.
Yeah.
So that filler right at the bottom right. There we go. Oh, we're good. But, slow time. Getting closer to you. Round down his 10.
Very cool.
021. He just released his nose out the front. Tilt down a little bit. Zooming to that.
Looks like the siphonophore dropped something. I don't know what it was.
Some right side.
I'm trying to get a closer zoom on it.
That's it. Best advantages. I'll do a plus flyby, and then it's time to go. Yep, I'm looking straight down. Okay.
Great shot, guys.
Nice work, man. Nice fun. Ace. All the basements. Descending to the middle, the left, right back to 57. What again? Oh, okay. Don't worry about that. There we go. Zoom in. Last line. Alright. That's really good.
Looks like we get a good zoom in on another Tina-4.
Use that guiding sight you can't really.
And it was just scooted inside our light. See if we can back up real quick.
See, my focus is like I haven't even.
Yeah, that's nice.
You went down a few meters from that. All good. End 800-meter transect.
And that's the end of the 10 meters at 8, or 10 minutes at 800 meters.
What's that guy on the right? Yeah, good.
We're going to go get
Can
some
we see what this is
extra
before you
credit
leave?
here. Yeah.
Look at that one.
maybe not.
Looks like another zoom on a different type of siphonophore.
Yeah, that's good.
Setting here.
That's beautiful.
Get you out front again.
Hi, this is Taylor.
Start now.
Yeah?
Hi, do you know much about these types of siphonophores?
Well, I know who to send to. They're all Physonex, but I'll probably ask Marsh Youngblood if he can identify them.
Okay, yeah, they're so beautiful.
Let's try rolling with those reds. You ready, Tom? Hammer down. Let's go. 45 down. Let's start.
The vehicles are just starting their descent down to 900 meters.
And we can include that red thing with the 800-meter transect. Oh, roger that. The red thing was important. It was 803 meters. How's the Lieutenant looking, Paul? It's alright. Getting below me. Speed up a little. I just bumped up to about 80 or 85% now. It's not a race.
Well, everyone who's joining us, thank you for tuning in and watching the Okeanos Explorers live feed from an ROV dive in the northern Gulf of Mexico. Right now we're conducting midwater transects every 100 meters from 600 meters to 1,000 meters, doing 10-minute runs at 600, 700, 800, 900, 1,000 meters at 0.3 knots over ground. What you're seeing right now in feed 1 is the HD feed from Little Hercules, our ROV, and feed 2 is the high definition view from our camera platform Sirius. You can see Little Herc in the lower section of feed 2 as they descend from 800 meters to 900 meters. Little Herc is just passing through 840 meters right now. Both vehicles heading a little south of east. Little Herc heading is 102, and Sirius is looking 099. In feed 3, you can see our scanning sonar. The left-hand sonar is from Little Herc, and the right-hand view is Sirius's scanning sonar.
Oh, 30 out front. This guy slow down a little. I can speed up. I'd rather dive below me though. No. Oh, far below you. Now.
All right, everything looks good, and the vehicles are starting their 900-meter transect.
Quick zoom here.
And a shrimp coming into view a little hurt. I'm going to try and get a quick zoom on it. Got its antenna out, or it did until we scared it.
I think so.
Definitely fewer copepods and amphipods at this depth than there were at 600 meters.
A jelly object on there? Yeah, that would be it. Jelly and a zoom and follow. Say again, Ron? It's a jelly. Do you want me to zoom and follow?
Looks like a jellyfish is just coming into view. We're going to try and catch a quick zoom. Oh, maybe just a mucus net.
Oh, look at near screen here. Got an amphipod.
The little amphipod or larvacean coming into the near field just in front of it. Or actually, it could even be a ctenophore. I'm not sure.
You got that one? Yep. Alright, let's come on. Yeah, the amphipod came next to it. It was actually swimming. Okay, the camera's back to 7 degrees.
Oh, I just got it back. It's a mucus net.
Excellent. Mike, did you see the thing that swam in front of the mucus net?
No, my internet's coming and going.
Oh, dear. Well, we've got all the stuff, everything recorded in full HD quality, and we'll make sure you get it.
Okay.
Another zoom here.
Another quick zoom on what appears to be a ctenophore.
Sorry about that.
Yeah, it's a larvacean.
Oh, correction, larvacean.
It's the animal in its mucus web. You can see that it's generating the current, the feeding current to move water through the mucus web. That was good.
Something centered on the right. Guess it was slightly above. Something coming in now. Center. This one, I think? Yep. Go for it, Ron. Jelly.
Looks like we've got a very small little jellyfish. Things that only our video engineer can see on his superior monitor.
It's teeny tiny. Alright. Full life.
Yeah, good job.
Any chance of getting a quick zoom on Sirius's camera? Got a shrimp in Little Heart view, and we're gonna try and get a zoom on some kind of almost string-like material in Sirius's view.
That's all I got. Coming back
Yeah, I don't know what that is.
I like this photo filter. Go ahead, what's up? Thank you.
I think it was a siphonophore, but I'm not sure. Nice shot of the shrimp, though.
Directly below me, but a little bit more time. I need that 4 meters. Alright, well, we got about 10 seconds. Anything you want? Alright, just that. Okay.
Very good.
Got 2 hands, 5 fingers. Good. That was pretty impressive.
In order to make those last two shots possible, we have one video engineer running both cameras, and so he had to simultaneously zoom and focus both cameras at the same time, and iris, and adjust the blacks and colors. Our super video engineer, Roland Ryan,
How many
ladies and
hands does
gentlemen.
he have?
He's got about 5.
People need it, special user. We'll leave these that good, just ask. Okay, my camera's back to 57 degrees. Depth looks good. Top left. That was a nice tag team there. Remaining.
Got about 1 minute left in this transect.
Oh, it's yours.
And zooming in on what appears to be, I'm not quite sure yet. Probably a larvacean is what, or maybe even a ctenophore, not sure. And I don't think we're gonna get it.
That's my fault. I'm gonna blame it on the jelly. It wasn't cooperative. Begin 1,000 meter transect.
And we are starting the transect now at 1,000 meters. 10 minutes long, doing 3/10 of a knot through the water column.
I got a something wrong. Looks like a fish. That would be a fish.
Something's coming in, looks like a fish. Fish is oriented vertically with a head down orientation. Working on a zoom now. And there it goes.
That's all we got. before he skedaddled.
As the vehicles make their transect through the water column, and we do see a few copepods or amphipods dancing around as they get disturbed by the pressure wave created by the vehicle. Looks like a small jellyfish coming into view. Gonna try and get a quick zoom on it.
He's like, what's going on? Yeah,
And he snuck away.
Sorry, I ran out of minutes. Lost for shit. Am I going over there?
Looks like another mucus net. Getting a quick zoom on it.
Yeah, I'm missing most of these because the internet keeps dropping out on me.
That's too bad, Mike, but we'll definitely get you the high res stuff.
Roger that, Blake. That works for us because we'll probably want to reorientate the vehicles as well. So we will follow up with a bit. Good position there. So yeah, for the moment, yes. Right on point, a bit of touch up there. There's our sign. Let's get a quick shot on it.
Got something in the water column. We're going to get a quick zoom. Looks like either a mucus net or a larvacean.
There we go. Alright, that's all we got time for. Not a phone. Yeah, nice find in there.
And we're out of time and we got to move on.
Roll. Yeah. Catching up, Carl? Slowed in. FMA was 70 now.
So everyone at home will notice that the little Hercules time is very limited on what they can stop an image, because if they get too far behind Sirius, they run the risk of fouling the tether or getting pulled off station. So the pilot has to walk a very fine line of stopping and getting good imagery while keeping ahead of Sirius and the ship moving behind it.
I do think, though, that they've effectively demonstrated that we can image this stuff in mid-water. We got some really good images of some of the stuff.
Absolutely. A couple of the siphonophore images that they've captured have been outstanding this morning.
Well, I appreciate everybody up on it.
So we're gonna get to bottom and then do a shift change. So then now we're coming up and around. Go ahead. Slow, yes. It's going away from us.
Get something very reflective in the water column we're trying to zoom into, but it was very small. I'm really not quite sure what that is. People, it is. It appears to be some kind of
It's
fish.
a hatchet fish.
A hatchet fish.
That's what it looked like.
Yep, it is. It's got that kind of flat shape, but it looks very pink and somewhat transparent.
Yeah, it's a hatchet fish.
Yep, hatchet fish.
Alright, you good with that one? I'd like to get one shot really full frame in focus. Come on. Relax and keep them from time to go.
Very nice.
This transect feels faster for some reason. It does seem that way. Maybe once we get out ahead, try to move into the center of the screen to get a little farther here. It helps that close the hours till you get we just won't look. We'll see what we can do.
Just so you're aware, I've got two laptops here, and I've got one on the Ocean Explorer website and one on the Discovery Video website. And it looks like the Ocean Explorer one is more stable. I'm losing it less often than on the other one.
That's good to know. Thanks.
57 degree camera open. Copy that. We gotta find one more interesting thing.
Got about 1 minute left on this transect.
I wouldn't mind taking one of those rocks and imaging it, see if it's actually a quiver or if it's just snips. Copy that. Let's do that at the end of the transect. Arch, the ship would like to come to a cruising stop. When we're done with this transect. Once we're done, let's prepare our climb. I lost it. Let's pull that. Copy that. Passing out of all cameras now.
We
Hey, Rowan, look what I found.
encountered another object in the water column, and we're going to take a stop. Looks like it's probably a siphonophore.
Coming back up. There's nothing going on. The screen has no visual on you or the cutter, so. Copy
Yep.
that. Hold down to watch the delta. Yeah. It's pretty easy for me to look up. I got on these. You're gonna start pulling me at some point. Drifting out on your own? Yep. And that would be the sign that I'm starting to leave. Yep, we're leaving now. Regardless of whether or not we want to.
All right, got a nice little view of siphonophore there.
1,180 meters, and we have bottom 6 meters off. Now,
You're already pointing in the way we want to go. Alright, I'm looking straight
1182 meters, altitude is 6. Base reflectivity from Sirius. So you'll see that it'll stay at a constant distance from us while the rest of the bottom moves toward us, and so you get a sense that, and once we get over to that area, it's not there. It always stays out in front of us. So you may be looking at a reflection of Sirius. Just wanted you
Yeah,
to be.
we saw that in the Midwater survey earlier, I think, too.
Okay, great. Nice shot. So we have bivalves that are intact here. They're dead, but they're intact, which is interesting. I mean, they're hinged.
Would you like to start or wait for? I was gonna wait till Tom got in here. Alright, video while we're sitting here. You can check this guy out if you'd like, check him out. That's good, thank you for allowing. One more zoom. Getting him back in the light for, alright? Alright, go ahead. 012. I was thinking 0.1. It'll take about an hour to get to where we think the target is. And it'll allow the pilot to do some pretty wide zigzags.
Okay, our depth is 1175.
It's too slow, you can speed up. Okay.
Is Webb out there? I want to get the real time working. I asked him once. I think he's got a ton. He's working on it.
Good, gyrating up. Yeah, you can keep going in.
Nice job, pilot.
Okay, can we get a ship move? 223 meters, bearing 012. Oh, let's come wide video. At 0.1 knots. Yes, it is. Alright, Tom, 012. Copy that.
I mean, if it's more of a briny area, you might get that close to the bottom. I would think really that you're going to get. I mean, if it really is bubbles, yeah, you'll get more of a dust, a haziness in the water column. It's very clear right now, so we have a good, control image.
Let's do a quick snap zoom over here. Spot stuff, please.
That looks microbial. Pteropod shell. It has some fluffiness to it. Doesn't look like it's. You can almost see ovoid circles there, right? At the lower left, if you look hard. Reminds you of a sponge, actually. What do you think? Yeah.
Co-pilot, is that a patch of hard bottom and Sirius? Come on.
Yeah.
It's definitely hard blue bottom. I don't have a distinct shadow behind it.
Yeah, that looked almost sponge-like, not, I mean, like a hard gelatinous material. We'll keep a lookout for active seeping here.
Hey, video, you're going to be our eyes here for the cloudiness in the water. It kind of looks already a bit cloudier, but that's just me being my altitude and lighting.
So it may be too that when we do come across these stains like this, like you're doing now, we might want to just get snap zooms and then just to see if there's any activity at all. I mean, this is a seepy area, so if it's what's our current like today, guys?
Doesn't seem like there's much of it, actually.
Okay. See, this looks like microbial material. See the filamentous nature of this waving. In the bottom left, you see some ovoid shapes too. I don't know.
Yeah, I'm creeping out here. trying to peek in here for you. Yeah. I'm not seeing anything amazing. Definitely not seeing any, yep, motion in the water. Okay, let's come on.
Actually, now let's drop a target here.
Right here.
That's what, D16
Yeah. Take out that
dash 1. Just put out white staining. We'll just keep a log of where we see these white staining areas that we stop at. Yeah, you see those? Do you see little fleas jumping around on the bottom here, like we did yesterday? Yeah, I've seen some of them. They're just on that white staining. One's over one of the burrows there. Interesting.
There's been a couple of hard targets that have stayed where?
Yeah.
So the points stayed about 30 meters out. We're talking about this area right now. There's no shadows.
Ah, come on. Another little patch of brine and microbial material.
Go ahead, Peter.
Getting a snap zoom on that. Some broken shells, one valve intact. Yeah, so you can see those copepods jumping around, white staining. Yeah, I'm just seeing the copepods just hovering just above that white staining and dark material. Don't see any trails here from. There we go. Sorry. The two crabs together. I don't know if I see enough legs though to be two crabs, but I guess it is. Legs up. We'll let that go.
There's a fuzzy. Just blow the lasers. Fuzzy. White fuzzy.
So again, to
Oh.
left of the white, that dark thing.
Yeah. And there's been a bunch of these hang on. But yeah, go ahead. Confirming from the coral. Yeah. It looks to be a worm. Very long worm.
What is that?
Be a worm.
Does look to be a worm on the end of that.
Okay. Focusing on the tip.
It. I mean, it's almost like a pogonophoran worm. But how does it attach to the?
Look like it's inside of that.
It's coming out of that tube. The tube is covered with hydrozoans. Certainly at the uppermost part.
Sorry about that.
It looks like a pogo out of its tube, and I guess I just don't understand that. You can almost see the main sort of aorta, right, going down that red line.
You want me to come find you, Bob?
But the tip doesn't make sense. Can we just stay on this for another minute? I want to see if we get the tip in the field with that central artery, if you will. Worm, translucent tube, thin red body inside. Absolutely, that's what I see. I just don't quite understand. There's several of these that we've passed over actually. Let's take a quick look here because I don't. It looks like this worm is out of its tube. There you go. Is there any material on the body that's exposed here? There's no little projections coming off. There's a polychaete on the tube. I guess, is that just part of the tube? It's a translucent tube, perhaps, that it's just clear, and we're looking through the tube. If we can come to the head end again, the tip, and see if there's an edge of the tube, or what. This is nice. Nice job, pilot. Video. Oh, let's hand this over to Brian. Brian, what do you think here? He's furiously going through his invertebrate textbook right now, trying to figure out what that is. Anyway, just let's get a good shot of it, then we can ask others. It does look like there's a pair of eyes on the end of that. So my guess is that this is some sort of a worm in a translucent tube, but doesn't start out translucent. It may get colonized and covered over by things over time. It looks like it's broader at the base, right? It gets thinner as you go, but then it turns into this translucent part. And so yeah, maybe you're seeing the process right here. Is that you look up, if you look up slope of this thing, or look up the length of this worm, you see it actually may turn into a darker tube. So maybe this is just a progression of how it grows and is exposed. It's bizarre to me. Seeing some burrows here too. You can see how midway along it's sort of partially translucent, covered with hydroids. Yes, yes, please. Yep, there we go. Like right there. So it's semi-translucent. That's interesting. Right, you see the junction right there. Wow. Yeah, roger that. Nice job, pilot. Nice job, video. Thank you. Wow. You should. You should. Yeah, nice job, guys. Thank you. All right, here we go. Heading north. Our depth is 1159. What's our distance, navigator, to the target? Yes, the shells are much more extensive here. Now they're actually on top of each other in places, stacked up. Guess I've seen two morphologies, one more elongate. Still seeing some gray sediment here, like it's been worked a little bit. Those are, skylights in these shells. No big holes.
Yeah, go ahead now.
sonar. Abundant shells here.
Would you like to drop a target here?
Yeah, now let's do that.
Go ahead, zoom in.
There is a tube worm there. See, I think it does. There's one at the base, at the very base, at the interface of the sediment and the rock. Yep. And one of our squat lobsters there. This rock is heavily pitted. Look at that. You see all the nice little holes for crabs to be in. Could be some live clams in there. I don't know. You see some that are partially buried that you really don't know until you excavate those guys out.
Hey, Tim, it looks like mussel shells down too. Did you see that?
Can you say that one more time?
It looked like I saw mussel shells down there, little clam shells.
Yeah, I think you're right. Those are.
Real quick, and then we'll go to the right to the worms.
Yeah, pilot. We also think there's mussels down below here. Just want to see if that's true or not. When you're done with this guy, we'll come down right in front of us.
Stop the ship here. We're still 130 meters from the possible seep from the multi.
How much time do we have left if we don't stop?
Maybe another 5 minutes.
Oh, we're okay.
Okay.
We're okay. Hold on then.
You got a good one, video? Yes. Okay. Come on. Sounds like a bunch of worms on the right. Under next to lasers.
Roger that. You want to do a quick zoom on those guys?
Yeah, go ahead.
There they go. Some nice healthy tube worms. Nav, confirm you did put a target here?
Yes, this is a little bit wider. That's dash 2.
Roger that. Thank you.
Yep. You guys saw a shrimp if you just I'll also get the shrimp in the shot. Good shot.
I don't see any holes in the seafloor that would indicate any kind of methane expulsion or any kind of bubbles coming up. But certainly I've seen an active seep area here, indicative. And yeah, reported in that pile right there when you're backing up.
Better angle here. Good partial. Right.
Tyler, is this where you think you saw mussels down here?
Yeah, it is,
Could be
but
one.
now I'm actually not seeing.
Yeah, I don't know. Maybe the lower right there, but I think it's mostly clams. It looks like mussels, but I think those are clams. Oh, I don't know. Yeah, Pat, I guess the go 2 o'clock to the lasers right now. There's 2 shells at the top of the screen that are parallel to each other, laying side by side.
And it looks like the ones on the lower right are mussels.
Lower right? Yeah, how about where he's zooming in now? Go right there, pilot, on the lasers.
Go ahead, video.
The long ones there.
How are we looking, Tom?
I'm thinking clams. We actually got to
Sorry,
push
dang
on
it,
here.
They look like mussels too.
Okay, so yeah, okay. We're gonna push on from here and keep going. We still have another I don't know, more than 100 meters to go before we get to the seep area. So the bubble area.
Roger that. Thanks.
Still seeing scattered shells, but seemingly less local relief.
You got something 15 meters in front of you.
There's another sort of gouge in the seafloor there. I'd like to know what causes those. This otherwise seemingly undisturbed sediment. We've got these big, 10 centimeter wide or more, sort of a gouge in the seafloor. Some people often think those are beaked whale feeding activity scars.
East of the first one.
Roger that. See it on the nav screen. Perfect. That's what we'll do when we'll get up there then. We'll look around and then we'll head off to the east if we don't see anything. That's great. Thanks.
Tubes on the left, that rock that's on the left of the screen.
Sorry, how many meters nav? Sorry.
130 due east.
Roger. Roger, thank you. Can we get a quick zoom on top of this? Is that a coral up top of there or is that just a rock? That's just... It's a fish. What is that? It's a fish. It's a fish. Can we get some footage of this, please?
Go ahead, Julia.
Interesting guy there.
Do you have an up there video? Because I'd like to get closer, so I just want to grab the snacks in case he sprays. Okay, come on. Doesn't look like this guy is spraying very fast.
So coming to a seep area. Lots of shells. Once an active seep. I haven't seen any live tube worms here. You see tubes? Oh, I do see some down below, straight in the middle of the center screen. There's live tube worms underneath that ledge, and around the side of that ledge, and on top of that ledge, just behind this fish. Benthic angler. So we see the barbel up top, right? That little, see the fishing pole there? Just chilling.
Guy needs a hook.
Guy needs a hook, yeah. He's lost his tackle.
You see this guy on Star Wars?
Yeah, looks like an elephant's foot.
Be aware that I'm moving faster this time, Bobby. Yeah, roger.
Yeah, there's live tube worms there off to the left. You see those?
And also in the background as well. Lots of tube worms in this area. Yeah, Okeanos got a lot of tube worms up over there.
Right. Now let's put a target here. It'll be 4. Be live. It'll be active seep.
I was just going, leave it, you can leave it this but zoom in. Going right there, yeah.
Yep.
When you're done here, Bobby, I would jump up over the rocks and take a quick look at what's on the other side.
That's a nice shot. There's more on the other side, I think, too. We can look at. It's awesome. No, nice job.
Jump to the other side.
Absolutely.
Full round. Don't care whether I'm this fish on our way to the other side. Got a minute or two. Can I get a tight open fish? Yep, full crush your tail there.
Coming in.
I'm coming back to get above the other rock. Watching you. Okay, gonna have to go left, right.
Again, have not seen any corals on these rocks yet today. There's a bunch of tube worm bushes here. Those that are exposed and valves open. What is this dark material inside here? Those mussels? Those are mussels.
Alright, let's do it. Quick
Roger that. Um,
Yeah, we'd have to come back actually.
I'm looking for methane coming out around here. That's what I'm looking for. And no, we got it marked. We should move on. Get what imagery you can here, best you can, and then let's move on. We'll have it marked.
Did they get any piece of this? Yep, that's the mark. Time I'm gonna keep this.
Nice shot video.
My target doesn't get wrapped on GoPro. Actually gonna come back as well. I see the target. Okay, 'cause
So, folks, we got this site marked.
actually on that rock. Oh, you're close. Yeah, well, actually, we're just
It's all
10
clear.
minutes from the top target, so it'll be
Look at that. Interesting. We do have a. We'll have to look at this more closely here. We have some interesting potential targets here in this in Sirius's sonar. Here we come across some more white staining and dark staining. We're gonna get a zoom in here again as we do. We'll come to these places. Thank you, Taylor. Sounds good.
looks noticeably flatter in pilot mode.
Yeah, it's true. Well, I think we're coming to the top of this thing.
Yeah, I agree.
Yeah.
Well, it might be.
Thank you, pilot. Yeah, I think we're up here on the rim or edge here. It starts getting flat here.
The Seep target is 20 meters ahead of where the Hercules.
Of a possible seep area from Tom Weber. Thank you, Tom. And so we think there's release here somewhere in this 150 meter vicinity. I got time for a zoom on the coral.
Yeah. Trying to get a better
As long as we're looking. Right. Roger that. Thanks.
keep an eye out for any worms. I think I see a few worms.
I'm presuming, this is my first time hunting for methane bubbles per se. I have come across them before, but if I'm presuming that the streams would be so strong on the seafloor to be recognized by the multi-beam, so it wouldn't be that we're looking for a bubble or two here or there. We would need to set up on a tube worm area and sit like this for 5 minutes and wait for a bubble to come up. I'm assuming that we would see somewhat of an active stream. So now we're zooming in on this coral. We got an octocoral here. I don't know if this is a purple Paramecia or not. We're having a navigator change at the moment.
Yeah, videos. Yeah,
Yeah, we will. Welcome,
Hello,
Nav.
it's Mashko here.
Hey, Mashkor, how you doing?
I'm fine.
Right. Wait 1 second, Mashkor. I'll be right with you. Hey, Nav, can we drop a target here, please?
Of course.
This is 5. It'll be D16-5.
Mashkor, this is Roland in video. Just want to say hey.
Purple coral.
Hello.
Hi, Mashkor. It's perfect timing that you would call, and we would be on the site here where you and others of the multi-beam have documented methane bubbles coming up. We are currently sitting over the area right now with Sirius right on the target, and we're stretching out with the tether, with the ROV to different compass rows, looking, and also to hard bottom targets that we see in the sonar. We just come across one here and see a coral on top of it, all the while looking for any signs of methane release.
Yeah, the video is looking great over here, and excited also to looking forward to seeing the seeps. Holly Tom is there too, and so Tom, have you looked through the multi-beam data to see how many targets we are seeing?
Hi, Meshkor. I don't have the multi-beam data from that these guys ran last night, and I haven't had the chance to look at to pull up your leg one data. What I did just do is send Tim an email with a picture that shows where they are right now and where the seeps are that we detected last fall. There's 1, 2, 3, 4, 5.
People on shore, this is Tim, back in the chair, and Navigator, just have a question. We're about what looks to be, I don't know, 60 meters from the Seep target? 85 meters. Did you guys decide to go north?
We went
So we're looking, or?
north a little bit to see what was up this way. There was another set of rocks, which we're on the north side of now. We were curious to see what was up here.
Okay. All right. Seems to me, I don't know if, did the shore side, did you guys chime in at all on?
We were up in the water column. Trying to
looking.
look at the sonar.
Roger. All right. It seems to me we want to work between the two possible seep areas.
Agreed.
So why don't we come back, so where the ships move probably, right? Somewhere in I think they're about 120, 130 meters apart, which would tell us we can't.
30.
Okay. Okay. We sort of looked around the one on the western side. Why don't we go toward the one on the eastern side? I think so.
Okay.
I think so. Yeah. Okay. Thanks.
All right, new direction tells me 130.
Yeah, those of you shoreside. I'm assuming you can see the nav screen in one of your displays. I'm trying to see. Maybe you can't.
180 meters, 130.
No, I don't think we have the nav screen.
2 knots, please.
Tim, I'm not seeing a nav screen.
Okay, roger that. You've got the quad screen coming over. Okay.
Okay. Sorry, I missed your read back.
Yes.
Okay. Did we pick up targets based on the data that we collected last night, or from earlier, from previous data?
Roger. I have not seen the data from last night, other than what may have been real time, right? Saying that there was activity here. I don't think it's been fully processed yet.
Yep. There is also a little bit of leeway because sometimes you see a seep in the water column, and then you have to kind of like judge where the bottom part of it will be.
Yep.
So I found this, the first point which I'm saying is towards the east of our current position, about 400 meters. It's a very strong signal right next to the bottom. So that's why I think it's a pretty good target.
Okay. So from what I'm
Picking something in the water column, then we will have to trace it down towards the bottom, which may be off. It may not be coming exactly vertically up or down.
agreed. Okay. So what we can do, we got a 180 meter run here to this possible Seep target 2 that Tom gave us. We'll go over there and look.
Okay.
And then we'll take it. Do you have a, can you send us a coordinate, a lat long for your,
Sure.
the one that's 400 meters east? Because that'll be closer to get to from that possible Seep 2 area.
Yep.
So we got another possible Seep area coming up here. Abundance of clamshells increases right up to the margin of the rock. Don't see any tube worms, but lots of valves here. Again, making our way to the southeast to another possible Seep area. Okay, Moshkor, I see the numbers coming in now. Thank you very much. It's great.
Okay.
We're going to put these
Tom,
up.
do you have the access to these data from EX1202 leg 1? It should be on the FTP site also.
Yes, that's what this is, I believe this is what we've been looking at.
Okay.
To pick the dive locations for today. So there's 2 positions here you're giving me. And, oh, I see. The one's to the east, one's to the west. I got it.
Hey, Mashkor, what line are you looking at from leg 1? I'll run it through the automatic Seep extractor.
Okay, it's line 264.
264, thanks.
Hey, Nav, this is
I
Tim.
can give you the exact time.
I've got.
That's okay, I'll just run the whole file.
Okay.
So Nav, one's west, one's east, right? Okay. Thank you, Moshkor. We're gonna. Brian's gonna plot it up here, and we'll send you over the HiPAC screen and show you what we're looking at here.
Roger.
Roger that. Lots of shells on top of this carbonate slab. Depth is 118. Don't think I see any tube worms just yet, but lots of. Nav, I know you're really busy. Maybe a target here though, just to mark that we have a.
Here's the active area.
Right. Well, that's really neat. Lots of standing here. Yeah. Put a target. I think we're on 5, 6. Is this?
Yeah, 667.
Roger that. So we'll just call it a.
Something vertical here.
There's active Seep.
It's not a tube.
Big brown area. Okay. This is our largest area of staining so far. Looks like there might be some clams in there that are alive. I see 2 fish resting or sitting on the bottom.
Okay. Tim, do we want to linger or keep going?
No, I think unless we're seeing any traces of any kind of bubbles, I think we got to keep going. I'd love to come.
No bubbles. I'm looking.
Right. Yeah, I think we got some live clams here, though. This is a very large area of staining. Got one eye here on the sonar Sirius, which is 20 meters off the bottom. Don't know if we'd see hard returns of methane, but we're plotting in points and
Well,
shells with sediment inside. Seeing some relief here.
much more pronounced bonding to the tin.
Yeah, these are extensive mounds here. Can we get the
That's fine.
Can we get the lasers on, pilot?
See a little bit of haze over. I don't know if it's my imagination.
I agree with you. Did we come this way?
No, this is new territory.
This is just going upslope, isn't it? That haze may just be the sedimented landscape in the background, but this is a large area. It's almost like it's uplifted carbonate here, just big slabs of it. Lots of shells right at the margin of the base and on top.
Watch the points I plotted.
Roger.
The eastern seat is to the southeast.
Oh.
The other one, and then we are almost 400 meters from the western seat.
Roger that. Okay, let's okay. Video. Can we put the high-pack screen across for people just for a moment? I'll talk to them. Feed 3, guys. I'm going to send you the nav screen. Thank you. We're going to put the nav screen on so we can show you as we're looking at this nice Seep area here. Going to show you sort of the distribution of the possible Seep targets and what we're looking at right now, what we're doing. You'll get the nav screen in just a second here. Thank you guys for continuing to log. It's very helpful. I really appreciate it. This is great.
Sorry to use this video.
Is he calling you? He's calling you.
Hey, Art, in the rack room, like one rack facing towards the center of the ship, there's a collab drawing keyboard. You could go there and scroll out twice and go to high pack right.
At 25 meters.
Okay.
That's just the ledge in front of us. Roger. Roger that. Okay, folks, well, there's a nice hole there. A lot of staining around it. I think that material that's in the water column is something that we probably stirred up, I believe.
Yeah.
Right, it is.
Can't be.
This is a deep thing. Our depth is 1119. We're a half meter off. So those folks shoreside, you're seeing the navigation screen that we're looking at now. You see the position of the ship there. And we're seeing us at D16-6 brine staining. You see the possible Seep target down to the southwest. And at the same latitude, you're seeing possible Seep target 2. That's the one that Tom Weber gave us this morning. You see possible Seep target 3 to the southwest of that. Sorry, southeast of that. That Mosh Core just gave us. And then you look way to the west, to the edge of your screen, you'll see possible Seep target number 4. So we've looked at possible Seep 1. That's where we came on and found activity there, Seep activity in 2 berms near there. Then we've come up north and we did our little look with the sonar from Sirius, and now we're heading beeline back to possible Seep target 2. It looks like that it's only 50 meters or so, 60 meters to between Seep 2 and Seep 3 targets. So I am proposing that we move to possible Seep target 2, look around there, convince ourselves if we're not seeing something, in any tool that we have, and then we'll go to possible seep target 3 and do the same thing. From there, we can reevaluate with how much time we have. I suspect we'll have to come off the bottom at 3:30, which is you know, a little under 3 hours from now. We'll have to come off, so it's possible we might be able to run the possible Seep 4 and get a look at that as well. We certainly have an active Seep area, I think, at D4, D3, D4. We can also look at, but we're in the neighborhood. Keep your eyes out. If you see really murky water, see something in the sonar, please let us know. Any thoughts on the plan? If you want to suggest something else, please do. That's just what we're thinking right now. We're headed to possible Seep 2, and then we'll go to possible Seep 3 targets.
Them this Mashko, that looks sounds really good plan. I'm also wondering if we have any targets on
Shells here. Pretty extensive. As we're heading due south now on our heading with a course to the southeast. Very extensive. So if everyone's comfortable, I'll change the nav screen back over to the sonar so you can see that.
Do you want just sonar or do you want the multi?
Let's go to the multi. Let's do the quad. So we've got staining here as well. Lots of shells. Hey, Ben.
Just a moment. He said he'll be right back. In the meantime, if you need anything, just let me know. Hi, Dave.
Very concentrated area right there.
Yeah. Look at those mussels. Pretty dense here. Oh. Can we drop another target here, like D16-7 extensive muscles, I guess? Thank you. Okay. You can see the muscles are, you know, they occur along what could be those fault lines.
Two, check out 2, then 3, and then recess. Check it out. Yeah, this is cool.
Yeah. So, as I understand it, the plan for today will be, for the rest of the day, is that we're going to be investigating the 2 possible seep sites to our southeast. Looks like they're 40 meters and maybe a little more than 100 meters away. Those two sites. Let's see. Yeah, we just, okay. So you can also see tube worms among some of these mussels in the sediment. This is probably the densest populated seep area that we've seen on this cruise, this lake. And can we do a quick zoom on some of those live mussels? Just for a few minutes, some of the live mussels here. You can see that some of them have some sort of encrusting on their shells, anemones maybe, or I can't really see what they are from here, but I've also seen limpets that are mussel shells, or or even gastropods. Yeah, interesting. So there are two like little patches in front of us right now. We'll probably get to image both of them, do some close-ups, see what's growing on those mussel shells.
Okay, really? We're doing here.
Okay, there I am definitely seeing gastropods here on the mussels. Wow, this is definitely a first for me. I in the past I've always only seen limpets on mussel shells and not gastropods like this. But oh yeah, there's a chitin. That's right. That's pretty cool. Okay, so we got chitin and gastropod mussel shells.
Okay.
Okay, chitins and gastropods on the mussels.
Okay, tilting up.
Ah, okay. So Tim is very interested in seeing if we can actually look inside these mussels. So we might try to find a partially open guy and try to look inside of it. Now I understand that these mussels are probably Bathymodiolus, but I'm not very good at identifying them down to the species level.
Baby ones right at the top. Chitin's moving up there.
I wonder, does anyone out there know what the species of these guys is?
You see the baby muscles at the top of these? Yeah, I want to work this interior shot, just the angle's really good. Now I'm going to go left down because there's another one we can look into right there. Yeah, that. Oh, well, actually, they're all here. There we go. Let's just hang with that.
Okay, this one is open a little more. We can try to zoom in, but I don't know how much we'll see. That's max zoom? Oh, okay, there we go. Okay.
It's very interesting to see there's amphipods on the grooves of some of these
Yeah.
gastropods. You don't see it right here, but just above.
Yeah. Is that extending from the gastropod or
it
from the
is.
mussel? From the gastropod?
For the gastropod.
Okay.
He's turning right now toward us.
Oh, that's right.
See him?
Yeah.
Coming back this way on the margin. You can almost see where he's been crawling along the margin and kept it clean.
Yeah.
Right there, just on the edge, and you got that chitin just above, and the chitin is moving downward into the clump. There were also several anemones. There were also several amphipods on the swirls of those gastropods.
Another 15 seconds, guys.
Okay.
I'm going to tilt down a little, look at the...
Roger.
Hey, Tim, this is Marshper. Tom just sent out an email with an image from one of the earlier ROV work imaging a bubble sweep. So you can see it. You can check your email.
Absolutely. I've seen pictures like that, many of them. I know that they're very local like that. And it may be that we don't often see them like that. We often will see turbulence in the water column.
Alright, we're actually sliding into the C2 coordinates now.
Okay.
Roger that, Nav. Thank you.
We're approaching our first possible seep site.
So this is almost a continuation of what we saw D16-6. It really was a continuous seep area. You see these large crabs in here probably feeding live mussels, live clams. Guys, keep your eyes peeled for anything coming up out of the seafloor. Any murky water.
Yeah, I'm just headed in the direction of the highest terrain.
Roger. Yep. That's kind of the serious hit is on, is the bottom ridge right here is what we're seeing, I think, coming up. Sirius again is it's 18 meters off the bottom. So this is a big seep area right underneath what we saw in multibeam.
Little hooks, starboard quarter. Oh, just went out of the stop. Yeah, they're too close.
Okay, guys, we are turning to see if we've got something attached to Little Herc. It sounds like on the starboard side. Traversing over a massive mussel and clam field here.
Okay, let me just hang at it.
We are at possible seep target number 2. Really appreciate all your input today. Trying to locate a methane release off the seafloor. We're using sonar where we can, and also just visually looking out for bubbles. Methane could be coming out of any of these areas that you're seeing in your view, frankly.
I don't think we're going to see a seep near with the seafloor in it.
Correct.
Yeah. So, okay.
Right. So guys on shore, I don't know the exercise you did of pulling up off the bottom with Sirius. Wait, stop. There it is. Got
there
it.
it is. There it is. There it is.
Okay, so they are on the scanning sonar. You can see them clearly.
Yep. There they are.
There we go.
There we go.
Can I suggest a virtual target here or something?
Yeah, we will.
Okay.
Nav will
Okay, you can see it's coming right up and through that bunch of
stream.
mussels, right?
Yep. We'll come around the other side and get it. Come down the seafloor.
Okay.
Time is 12:57 local. Our depth is 11:19.
So if you got a really short range, Jeff, on the high frequency.
Did you put a yeah, thank you. The pin.
I'm working on it to average the position.
Okay.
It's that's pretty vigorous bubbling there from on the scales of what I've seen before. It's pretty good stream coming out. We're going to set up here and get some good video of this, good position. And see if we do see something, the ground truth, this observation on the seafloor.
Survey 2, Art, they're requesting you record sonar.
Tim, I don't know what's possible, but if you can kind of zoom in on the origin of that and with the lasers on.
Yeah, I believe we will.
Oh yeah, we're coming in. We're setting up now to maybe sit down gently on some bivalves here and take a shot of this.
Got a good shot in Sirius video.
That's really great. Nice job finding it.
Roger that. You look in the Sirius footage, you'll see Little Herc looking at the methane release on the seafloor. We're at 1,120 meters depth and 1 meter off the bottom. Confirmed methane release based on multi-beam data acquired by the Okeanos Explorer.
It also appears to be showing up in the EK69.
Roger that, Nav. We are also seeing it in the EK60 right now. Thank you, Nav. Got a little bubble here down to the right of us too, the corner of our screen. There's a little bubble coming
Oh
out every once in a
yeah.
while, just to the down right. But there is certainly a good steady stream coming up out of this raised clump of mussels. This hummock of mussels. The methane release doesn't seem to have changed the distribution of mussels. There's no clearing out. There's no large group just sitting over what's escaping. No visible impact.
What is that? What is the dark thing? Yeah, who lives there? I think that's the foot of one of the mussels. Okay,
You ready for a zoom?
yeah.
Okay, you guys see it in the Sirius footage as well, right? As we go in and see that bubbles percolating through the mussels. You've got chitons, lots of viscous thread attachments. Looks like this guy's been moving around a little bit.
Maybe even limpets on the muscles.
You can see the periostracum of the mussels pulling back. The open siphons. There's the gastropods here too. Remarkable to think you can see this in the multi-beam signal, an EK60 signal. Nice job, guys. So the possible Seep 2 target is an actual Seep, a very large Seep.
Switch from the nav on the quad split to the EK60. Would you like to make a switch from nav stream from the quad split to
Sure, video, if you can do that, that'd be fun. Is that okay? Yeah. And see if we do see something, the ground truth, this observation on the seafloor.
Survey 2, Art, they're requesting you record sonar.
Tim, I don't know what's possible, but if you can kind of zoom in on the origin of that and with the lasers on.
Yeah, I believe we will.
Oh yeah, we're coming in. We're setting up now to maybe sit down gently on some bivalves here and take a shot of this.
Got a good shot in Sirius video.
That's really great. Nice job finding it.
Roger that. You look in the Sirius footage, you'll see Little Herc looking at the methane release on the seafloor. We're at 1,120 meters depth and 1 meter off the bottom. Confirmed methane release based on multi-beam data acquired by the Okeanos Explorer.
It also appears to be showing up in the EK60 now.
Roger that, Nav. We are also seeing it in the EK60 right now. Thank you, Nav. Got a little bubble here down to the right of us too, the corner of our screen. There's a little bubble coming out every once in a while, just to the down right. But there is certainly a good steady stream coming up out of this raised clump of mussels. This hummock of mussels. The methane release doesn't seem to have changed the distribution of mussels. There's no clearing out. There's no large group just sitting over what's escaping. No visible impact.
What is that? What is the dark thing? Yeah, who lives there? I think that's the foot of one of the mussels. Okay,
you ready for a zoom?
Yeah.
Okay, you guys see it in the Sirius footage as well, right? As we go in and see that bubbles percolating through the mussels. You've got chitons, lots of viscous thread attachments. Looks like this guy's been moving around a little bit.
Maybe even limpets on the muscles.
You can see the periostracum of the mussels pulling back. The open siphons. There's the gastropods here too. Remarkable to think you can see this in the multi-beam signal, an EK60 signal. Nice job, guys. So the possible Seep 2 target is an actual Seep, a very large Seep.
Switch from the nav on the quad split to the EK60.
Sure, video, if you can do that, that'd be fine. Is that okay? Yeah.
Okay, they can see EK60 on the quad screen.
Okay, you guys should be able, short side, to see the EK60 on the quad screen. I hope that's big enough for you to get a sense of what we're looking at. If you guys want to see a full view of that EK60 shot, let us know. We could put it at full screen for you. Again, our depth is 1,120 meters. We're sitting right on the bottom, watching methane bubbling through a hummock of mussels.
Okay, that's come wide a little bit rolling. See that
Looks to be a well healthy clump. Seeing the formation of methane hydrates on the bottom of the shells.
Yes, I think it's a bar 10. That's the fire.
Go ahead, pilot.
Yeah, upper right third of the screen,
Roger.
pink object, far margin of the mussel. You see that feather in the center for you?
Yeah, I think, okay, I think that's, are they on the other mussels too? Like they're, oh, you can see the methane bubbles right there on the side margin of that mussel.
Come right a little bit.
Yeah,
Start up for you.
I believe those are byssus threads. These are the threads that enable mussels to move. They shoot them out of their foot, basically, like Spider-Man. And they,
What's the pink thing, Tim?
I don't, oh, I'm sorry, there's an anemone there behind it.
It's heavy.
It's moving.
Oh, it's a fuzzy wuzzy.
Fuzzy wuzzy?
Yeah, it's like a, it's a polychaete.
Ah.
I was going to say archinomi. Let's wait for it.
So Tom, this is Mashkur. I was just wondering, what else can we do here to figure out how best to pick up these seeps with our sonar? So now we have a seep detected. We can play with the different depths above the sea floor to see at what height we can pick them up, just for the future reference.
Yeah, you could do that. You could do the same procedure we tried earlier on possible seep site number 1 and just come up and see at what range you can pick them.
Being on the EK60, because it seems like we were getting strong hits and then we were not getting anything and then we were getting something. Is that how you are interpreting it, or?
Well, on the EK60, I don't know if it's the video feed, but I see a lot of noise in the EK60, but sort of behind that noise, you can see the big sort of red area, and that's all backscatter from gas. Probably the EK60 is at 18 kilohertz. It's probably
Thanks,
pretty
pilot.
close to resonant frequency for these bubbles that are coming out, which I'm having trouble getting an estimate of what size they are from the video here, but are probably I don't know, what are they, several millimeters?
Hey,
It's probably
Thanks,
pretty
pilot.
close to resonant frequency for these bubbles that are coming out, which I'm having trouble getting an estimate of what size they are from the video here, but are probably I don't know, what are they, several millimeters?
hey, Tom, it's Mimi on the ship. Can you hear me?
Yep, we can hear you.
So the red, distinct red lines on the EK60 are actually the bow thruster, and we're trying to cut through that noise with a longer pulse length. But unfortunately, we can't really get around that very well.
Yeah, no, I think, yeah, definitely you get bow thruster noise and the long pulse. It's a great idea to try to mitigate that. You can still see what's going on, at least if you're used to looking at noisy-looking EK60 data. So, you know, even with the bow thruster on it,
Sort of like quantifying what's on the bottom
this
first.
Is enough,
Yeah.
This is enough gas to cause a very big signature in the multibeam, but there could well be more gas there.
Okay.
I'm not arguing that point.
Okay. All right. Roger that.
So Tim, are you thinking to do like a 50-meter grid pattern around this site, or I mean, are you?
Yeah. I mean, we
to go to the possible Seep 3.
we basically come into here and just drove across it. I don't think we know the bounds of this at all. I don't think we know really everything that's here, right? We just kind of meandered into it and then saw this, and so so I don't know. We could do sort of grid patterns and look for the methane, which is be fine with me. Do some near bottom surveys and look out from here. So that'd be fine with me.
Yeah, and And Tim, do you see an associated live clamshell correlation with these seep? I mean, should we look for an area around that where this seep has effect on? I mean, what's the effective footprint of the seep in terms of life?
The effective footprint of life is tens of meters, as far as I know. Maybe every direction. We came on this about almost 80 meters ago. We started seeing sort of seep areas, and 50 meters ago I think we're picking up live things. The seam of mussels we saw along a ridge is good 35 meters away. So we've had active and live animals for a while here. It's a very large area. But in terms of local right around the methane, I don't see any compositional difference in fauna here where we've seen the methane come out. They're just if it shut off right now, we wouldn't know that there would be something there. So I think we should look around here a little bit more and see if we see more methane streaming out.
I see it. Just kind of back.
Yeah, Tim, this is Mashkur. I agree fully with you. And as I said earlier, my only one of my interests is like we can figure out how can we best use the sonars on the camera sled and also the ROV, so to better enable us to figure or to detect these seeps in future, especially during leg 3. We will be doing this kind of work a lot. So that experience will help us in understanding how best to use these sonars.
I'm going to rise up a little bit, come way down on Little Hook's range, so we just see the plume in front of us. Notice that you just see the plume in front of us. Come down to like 6 meter range on Little Hook, please. Okay.
I'm sorry, hey Dave, pilot, this is watch lead. I think the idea is not to have the marker even be that close. I think you want to be further away. But I'll let Dave talk to you about that. So shoreside, you're hearing this conversation, I think, about how we want to put a mark right here to make sure we can one day, if you guys want to come back here, others want to come back here, they'll know exactly where we were relative to the methane. We'll move off somewhere, some safe distance, so that the vehicle can come back, or any vehicle can come back and work without getting prevented by the marker. So the idea now is that we're putting the lasers on the bubbles that are coming out. Get a sense of the width of the stream. You can see it's we're heading 336, and you see it's leaning. What's that? Yeah, go ahead.
Sure. Are we capturing Sirius video as well? Yes, we are. Okay. Somewhere between your legs. Yeah. Just going to keep rising with it. Walking. Any tether problems, Jeff? Okay. 4 meters off bottom.
We are currently 4 meters off bottom looking at the spread of these bubbles. Not sure I can see the lasers actually. I don't know where they are, but maybe below. But it's 4 meters. We're now 4.8 meters off, 5 meters off, and you can see there's quite a spread here. Again, I don't see the lasers in the here, but you can certainly see the spread in the Sirius footage across the lights of the vehicle, the width of the vehicle. Our current heading is 353. It looks like they're coming toward us. I would say now we're at 7 meters. You see they're migrating in our direction, and we're heading at 338. So these things are going to the southeast. We're at 10 meters now, and it's clear they're being spread out, and I don't know if part of our thrusters are impacting some of that or not. But the reality is they're spreading out pretty rapidly, even at 10 meters. 11 meters off now, we're losing the bottom. Still have a view of Sirius in front of her. See the bubbles just. Now we're at 13 meters, 14 meters off bottom. Again, we're at our Seep target number 2. We're located in an extensive mussel clam seep field, very large with gastropods. 14 meters. Now we're at 11 meters. Still trying to see the lasers. And now we're coming down. We're at 8 meters now. These bubbles are really more or less going to the south, southeast to southwest. As we're coming down to the seafloor now, we're at 6.5 meters. You see we're driving ahead now, the distance of that coverage to the southwest, southeast. It's maybe taking up a current here in our direction. Altitude is now 3 meters off the bottom. So lots of mussels and clams, I think, organized on small faults, fissures, and now we came across the methane bubbling up, and been experimenting here with sonar returns off these bubbles, going up and down. Now backing off, seeing which point this the sonar signal is lost in Sirius. You can certainly see a little herc on your screen. You see the bubbles coming up pretty well. Yeah, the cursor's on it right there. That's a nice signature on little herc. That's a 6 meter range though, max range, so you can see 4.5 meters away, or 5 meters away, 6 meters away. Changing the range here to 10. Now we're at 7.5 meters. 8 meters. 9. So we're picking up some of the return here, and Sirius at 10 meters away.
horizontal. Just bump right a smidge. And there we are. Rolling, can you focus bubbles? That is focused unless I zoom in. We zoom in, let's see if we can figure out what our focus distance is. Just going pretty fast there. Okay, yeah, come wide. There's the focal range twice. Yeah, just can't tell.
Dave, would you do one more thing for me? Could you rotate to your left? And what I wanted to do is get a view with from Sirius to show the spacing between the front of Little Herc and the bubbles.
Yeah.
I'm trying to get us.
Did you see the shots we did earlier?
I saw some of them. What I'm trying to do is get a sense of what the framework that we're going to put on Little Hercules is going to look like.
If it was here now?
Yes.
Okay, so
Yeah, thanks.
So, Rella, we need some zoom on Sirius cam, please. Could you do my adjustment? 45 minutes opposite. Just to get the best. So, Dave, the bubbles are coming up right between my lights.
Yeah, no, this looks good.
From here. So, I mean,
That's about. There's a couple adjustments on the frame, but that's about the distance. So if you set down and just hold down a little bit, little downward thrust, and then tilt your camera up to horizontal.
go gently here. I don't want to bust up anything. Zoom in more serious. Stable. Waiting on the bubbleator.
Okay, so what's your camera angle now, Dave?
-7.
Could you bring it up to 0?
Yes. That's my full up location.
Yeah, see, that's going to work good. That's fine. So the center of the grid will be center of the picture right now. And it's about a foot square. So you should be without even adding any additional lighting or blue lighting or anything from the side, we're getting a pretty good image of the bubbles. Now, Roland, if Dave, you can have Roland zoom in a little bit slow on the bubbles.
Yeah, come in slow, Rolly. I'm curious what those bubbles look like in a still shot, because there might be some significant smearing. It's going to be smooth.
Hey, Dave. Hey Dave, can you go off of OKEX for a second? Let me talk. I want to talk shoreside. Thanks. Okay, shoreside scientist. They're right now looking at these bubbles and thinking about upcoming work they got to do. It seeps like this, methane bubbles, and as well as documenting what we have here today. I guess I have a question for those who. This is an enormous site, and they're working on these things here, and I'm thinking about the next steps. I think the idea here is to do this, and we'll roll off a little bit away here, maybe 10 meters, and deploy a marker that they figured out how to deploy. We deployed it for a first marker yesterday, and which is great. And I think that what I'm thinking about now and mulling it over, we have some other possible seep targets, but I think we'd also like to, now that we've found this here, maybe we want to push further to the southwest, okay, and see the edge of this field and make a contact with the edge of it. And then I would suggest coming back into it, either on a parallel line to the northwest or due west, and try to pick up sort of the margins and other aspects or features of the field, and then work the field a little bit more. Look for more methane bubbles, and look at the fauna here. Um, you know, I've done some seep work in the past, but not. I haven't seen anything as extensive as this before. Maybe others have. So yeah, I'm off. Sorry. So anyway, I just wanted to get some feedback from shoreside, you guys. I would be very happy to do that and document what fauna are here. We're noticing that we don't see squat lobsters, we don't see shrimp here. Be nice to have more of a look-see around on what's going on. It's nice to see amphipods on the coils of the gastropods and things like that, distribution. So
I'm still having a hard time hearing you.
Taylor, you having a hard time hearing me?
Yeah, I can't hear you over the pilot.
You shouldn't be hearing the pilots at all. The pilots are not talking on OKEX. Taylor, can you hear me?
Yeah, I can hear you now. Go ahead.
Sorry. Woods Hole, are you hearing the pilots?
Hi, Tim. I was hearing the pilots up until just now.
Okay. So I'm trying to just, while they're doing this work right now, a little bit of engineering work and looking at the bubbles and their distribution, I'm thinking about what we do next. And I was just thinking about coming to the southwest on the same line we had. Yeah. And finding the boundary of the field, and then looking around there, coming back into the field, and looking at some biology, and certainly the substrate they're on and all that kind of thing. So just looking at the field more. To my mind, this is a very large field, and I don't know if I've ever seen a seep quite like this. Maybe I haven't seen a lot of seeps. Taylor, you've seen more than I have. So I don't know
Yeah, I think it'd be a great idea to go around and look at the boundary. It looks like an amazing, extensive deep site. I'm really interested to see all the way around and then come back into the center.
Yeah, I think that would also benefit the mappers and others who want to know more about the methane here, and there may be more sources of release. So it may be, folks, that if we continue that southwesterly course, we've got 30 meters or so, maybe 40.
A little bit east-northeast.
The possible seep targets, I got two. We're sitting at the one that you gave us, Tom, and then just 30 meters or so southwest of here, we've got seep site, possible seep 3 target that Moshtor gave us. He gave us 2 points with the sonar that the confidence is not as high, and so I think as a method for the future, using little herc a little further off the bottom. May be advantageous for using the sonar to locate methane bubbles. Okay, as such, we're now about 0.8 meters off the bottom. And we're now moving off to the west, or westerly, northwest, to place a marker. So we're going to put a marker right where the lasers are now. You see that big
Hi, Tim.
hummock?
This is Julie.
Hey, Huey, go ahead.
Hi, we're still having interference problems with the pilot. When you're
Okay.
speaking and the pilots start speaking,
Okay.
they drown you
Wait.
out.
All right, wait one, Huey. We'll work on that. Okay, video. Okay, Woods. Oh, yeah, yeah. Okay. Okay, Woods Hole. This is Tim on the EX. You should make sure you look at your channels and make sure you don't have anything that could be open to the pilots. That's the only thing we can figure out here, is that there's some channel that's open that you're listening to, because right now, if you're on the OKEX line, you should not be hearing the pilots. Should not be any confusion.
Okay, roger that. I don't have any of the channels on that would have the pilots, except
Yeah,
the OKEX
we
line.
don't understand that unless the conference call. Are you on the conference call as well, listening?
No, I'm sorry, it's coming through the RTS, but I am on the conference call as well.
Roger that. Okay. Not sure why that is. Stand by. And then we'll come off the floor here and put a marker down. There's a tube worm. Yep, there's looks like a zoarcid fish. Okay. Thanks, Dave. Okay. Yeah, see smaller mussels there. So now you're looking at the snout of a gastropod with a mussel that has colonized its back. There's a small, I'm not going to say it's juvenile, but I'll say it's a small mussel, recent colonist. There's one, looks like just above the snout of that gastropod, moving over the mussel shell. In that crevice, you also see a recent colonizing mussel. It's right on the hinge there, it looks like. There's a couple of mussels on the back of that gastropod. There's also looks like to be, is that an amphipod near that snout? There's a red material. Maybe that's part of the gastropod snail. But you see the foot is extended, dragging over the shell surface. Depth is 1119. We're looking northeast at this seep area that we found this afternoon. Dragging over the shell surface. Depth is 1119. We're looking northeast at this seep area that we found this afternoon. This is a rather rare sight, I feel like, to be able to have this kind of imagery close up of a crawling snail.
Just picked up our scent, Tim. I hope it doesn't charge.
It's coming our way, that's for sure. That's great.
I don't want to blow in the calories. Well, Sam, yeah, I'm just letting this roll for a little while.
Yeah.
It's really sweet.
Little gaped open there. Let it siphon out.
Nah, this is fine, Rory. Let's just let this go just a little bit longer.
There is one at the base behind it.
Come left just to, it'll be pushing focus. Coming left now. There he is.
So you're seeing some live seep tube worms here below the mussels that you don't see when you're just flying by or getting a non-macro view. Pretty small. We're confirming the lasers are on. They're probably below. That's fine. I just want to note that they're on. We back up. We're way in. Roger that. I don't. And I just didn't see it. Maybe we'll see it back in the video of the stills. But a lot of small mussels here. Looks like this place is being actively colonized or has been recently. You see the open siphons there. Small mussels on the surfaces of larger mussels, sometimes in the cracks, in the hinge cracks area, and a lot of bissus threads you can see. These look to be rather healthy looking. You see a small mussel there at the center of the screen, surrounded by bissus threads, and here's a chitin on the left. These guys just patrol these shells, probably to eat microbial debris off. There is. There is. There's all this nice one at the bottom of the screen. There it's moving. There's a bigger one just below that chitin. Might see the light or not. There it is. I don't know. I think that's an isopod. Saw 3 of those there. More snails, bissus thread attachments. There's a polychaete at the right-hand side. There's a polychaete going down. How can you not love deep sea biology? How's it possible? Just on the sediment here, we'll see what Bob Carney thinks of this. This kind of waving out there. Let's take a look. Oh my gosh, it's a yeah, yeah, I'm trying to think of the polychaete oh my God, maldanid? Amphoretid. Yeah. I don't know why I thought maldanid first. Maybe not with that I don't know what this behavior is. I don't know what that's all about. Okay, maybe that was a last gasp.
Okay, let's come on,
Again, Nav, you said that marker was M28. Was that you who said that? Oh, sorry. Co-pilot, that was M28?
Yeah, M28.
Okay. Thank you.
Hopefully we'll get a good look at it in a second. Okay, so I'm looking right at it, but I can't see it visually, but I've got it in sonar. The bubbles here, yeah. Yeah, got the bubbles, so I'm going to rise a little bit more. I can see the bubbles in pilot cam.
Bubbles and pilot cam. Yep. So our heading is almost east, 116, a little bit south of east, and we're going to be deploying the marker. You can see it in the Sirius south of east, and we're going to be deploying the marker. You can see it in the Sirius footage.
Video, can you zoom on Sirius?
What he's got to do here is he's going to point the camera down, and when he does so, he's going to hit a pin. The pin's going to set a release, and there goes the marker. Didn't get much time for a drum roll on that one, but that doesn't matter. It worked just as well.
No fanfare.
Nice job.
Okay, we can read
Come on.
28. There we go. And I'll line M28 with the bubbles. We'll take a bearing.
Roger that, thank you. We're going to take a bearing here from the marker to the bubbles. To this ridge imaging a second ago. Depth is 1118. My event log is frozen. There we go. You got him, Mel? We're lining up in the sonar at 5 meters away from the bubbles.
115. Yeah, about 5 meters away, according to sonar. 5 meters, 115.
Thank you very much, pilot. This is a very extensive field of mussels.
Video, there's a plane straight ahead, just off to your left now. I got it. There it is. Visually, you really got to come right onto these things.
You do. You see it now in the center screen, folks. There's the bubbles leaving the seafloor.
Camera call.
We're going to look around here.
Video, can we go ahead on Sirius? Zoom it back in, but center it up. I'm going to light them up one last time, and then we're out. Go half zoom on Sirius. Thank you. On you, Jeff, but I'll be moving soon. Who's coming right at us? Wow. Zooming. That's great. I was drifting away.
There you go. You got him. You got him. He's even going to turn for us. Go up in the lights. Wow. To me, this is a very large site. I mean, it's almost continuous cover of mussels for at least 30 meters, I think. We're going to strike out now to the southeast to see just how far it goes. It may go all the way to the possible Seep target 3 that we had here. We're not seeing much in the way of crabs here at this site, with the exception of this guy here, this Chaseon guy.
There's another one just to the left, too.
So pelagic octopods won't feed on the bottom. So the Cerianthus and all those won't. They float around. Yep. Okay. Again, we've got a ship move on. We've got Sirius starting to move, and be headed to the southeast shortly. We're just looking around now. This is almost due east as we just look around the field. Lots of empty shells here. This is an expansive field. Mussels. Maybe some live ones down there now.
Thank you. If we're looking for more bubbles, should we keep the vehicle up? Then we'll kind of lose visual, good visual at the bottom.
Honestly, the best bubbles that I saw, visually, I saw in pilot camp first.
Before the summer? No, we saw them in Sonar first. Do you want to stop and look around here?
Yeah, no, I want to keep punching through.
Copy that.
Yeah. So we're pretty high up with Herc right there, and I'm not seeing any
We've got a
bubbles. Yep.
Hey, Tim, the Sirius sector scan seems to be frozen on my end.
Hey guys, is the holy cow? Hey, can we put a nav target down here for live mussel field? This is enormous. Co-pilot, is a Sirius sonar stuck or anything like that? Is it working?
We just had them on the same frequency, but I can switch it up.
Yeah, Tom, we just had them on the same frequency apparently, and that's
so
what.
I wasn't running both at
That's
once.
okay. So he didn't want to run both at once, but there you go. Nav, what'd you call that?
North edge of live muscle field. I can give it a number too if you want. It'll be D16-9.
D16-9?
Yeah.
Is anybody impressed by this, or am I by myself? I feel so alone out there. I think this is enormous, so I don't know. There's a nice little mussel lake right there. There's some briny area here to the left. Yeah, it looks extensive to me.
Tim, this Marshburg again. So this is, these mussels are based on that seep, or what
yes, this whole area is sort of percolating, or I would think radiating some seep nutrients, right, for the animals here at Marshcore. So you have live and dead animals intermixed. Like you just came over a nice large patch of live mussels that were clearly feeding on seep material. Now we're over dead shells that were places where it was once active. So yeah, this whole place we've been into for the last, I don't know, 100-meter stretch or more has had signs of activity in it. So it's more of a zone of seepage rather than a pinpoint island of a seep, if that makes sense. So we could go through this dead area here, let's say, and come across another live area with bubbles coming out, just a few meters away. If that's what you're asking, if that makes sense.
Yeah, that's exactly what it is.
Okay. Yeah, see, it's just kind of dead here. A lot of open valves. Not much in the way of staining. It's all modestly the same kind of color to the sediments. See a crab over here, maybe picking up some dead material. So we've extended through possible seep target number 3. We are getting some return in Little Herc straight ahead. This patch right here that might be it's a little slopey here. What is this thing?
This is a part that's been thrust upward, I think.
This is a cleared area for some reason. Something, piece of carbonate that's got an edge here. So it's a thrust up portion with a flange on the edge, kind of an edge to it. And lots of more bivalves on the other side. Don't see real live patches like we've seen before. But this is continuing on. We're looking to the southwest now.
You definitely see farther from up above.
Yeah, we're just doing a spin here. You can get a sense of it's just littering the seafloor with shells. Now heading east. Coming back over.
So we're dropping back down the hill here? Correct. We should be going down it slightly.
Heading is 150. Doubt we're going to see more activity out here, but curious as to what's out here. Here's another fish. My event logger is frozen at the moment. So depth is 1138. Turn it back to the northeast a little bit.
Let's go ahead and add 30 more, 30 more.
Feel like we can give this a little while longer here to see what's out here before turning back toward the field. If you guys have other suggestions, let me know. Or firm compacted. I did. I don't know. Okay. So getting a. Okay, back up. There's a ship, another one going across the field of view. I think we're getting out here. Things are really thinning out. Nav, how much more till the ship move ends? I know you put 30. You put 30 on, I think.
I did. Hold on.
So we're back at 171. Depth is 1141. Starting to see some mounds now. Small mounds. 20 meters high.
You just want to let This one expire?
I'm debating. I think we're out here. I think the balance would be much more excited if we moved on back in again. Fish out here. All right. It's a large hole here. Is that a hole right down there? A fish burrow. There it is. I don't know if we can see down in there or not. Is somebody home? There's a fish in there. Okay, so we've started to come into mounds here. I think that if no one else objects, we may call this the boundary. There's certainly maybe one or two of those transparent tube worms we saw at the beginning of the dive. Okay, heading is 043 presently, striking out toward the northeast while we move course over ground with the ship to the northwest. Standing out, just looking, seeing what we can find, see what we see. Depth is 1141. Coming back up slope. Certainly coming over shells with different morphologies. Looks like a couple of these bivalves are actually still hinged. Seen a couple. Striking out now at the heading of 300, 285. Little Herc is out in front of Sirius.
Look at all those seeps in the Sirius sonar.
Yeah, I know. That spiral pattern in the seepage is amazing. Those are all bubbles coming up. Look at that. Whirling around.
Okay. I also match score. I'm not sure what the current profile would be in that area. We don't usually see the seeps come straight up though. Yeah.
Yeah, Nav, what's the scale on the bottom left of that Nav screen? I can't. Is it 20 meters? It's 20 meters. It's just sitting on top of a label. That's okay. It's the top of a 20 meters. Aye. So let's say we're about 50 meters or so from where we saw that large lake of mussels, live mussels. 25 meters north, and we're heading as 052 right now. As we sort of do a zigzag back and forth over the bottom as Sirius gets pulled to the northwest. Almost due west. About 35 meters south of the that live mussel lake. Looking up slope still. Seems like the active part of this Seep is really on top of the slope.
Does he know you're there?
Wind changes. It hits you.
A lot of these bivalves are still hinged again. Seeing a few mounds. You can see the shell distribution in the Sirius feed. Looking down at Little Hercules.
Watch the map. We got about an hour bottom time left.
Roger that. Okay. You're keeping the ship on the move?
Ship is still moving.
Roger.
Tim, this Mashko, did we decide like we are going to stay in this field or try to head towards the western field?
Okay, Moshkor, we just went over to the possible seep number 3. We moved on past that to see if it continued on. We ran out of seepage and we came back. Yeah, we'll get a quick zoom on the urchin. We got time for that? Are we okay? And Moshkor, we'll come back into the field we know to be active. We thought we'd look around here for, we'd survey for both methane and for animals and features that we see. So we thought we'd spend some time here. Did you wanted to go off to another field, the one 400 meters to the west?
No, I was just asking because we may not have enough time to characterize that site fully. So my suggestion would be to stay in this field and look more here.
Yeah, part of the issue was that before we just had a discussion here in the van, we had 45 minutes left in the dive, and I felt like we'd had no time to get over to another field. I thought we'd just stay here. We just got an extension of an hour and a half, so it gives us a little more time. Thanks, pilot.
Do you think we will have time if we go towards the western field? It's about 700 meters away. So.
What it would mean is that we wouldn't look at this field and look for more methane and see if that's where this signal's coming from, if it's just that one stream. And I don't know how much time it would take to get over to the other field. I can find out. Wait one. Nav, this is watch lead? Roger. And my initial reaction was is that we stay here and do a good job of seeing what this site is about, instead of seeing if there's more methane here, seeing what animals are here, and ground truthing that.
Roger, Tim, I agree with you.
Yeah, okay. Thanks, Moshkor. So we're working our way toward the field. We're just south of the marker 28 marker. And the ship is holding, is that correct?
Negative. Ship is got about 20 meters remaining.
Roger that. 20 minutes remaining. 20 meters remaining. I think I'm losing it back here. Some dark material there amongst the bivalves. That's interesting. Taking a zoom on the sediment. Just this darker patch here. It's okay. No, this is great. Okay. Depth is 1120. We staying on the move here. Where ship's gonna slow. It's gonna stop its move pretty soon, and then we'll be able to look around at the Seep site. Chasing on crab, lots of dead mussels. Not really. There's a big mussel area right here that's really nice right here. So there's marker 28. There's the bubbles are here, right? And so we're coming to be around the. Yes.
I think I'm beginning to see
That's right there. Nothing.
on little hook sonar.
Should be. Yeah. Okay. That sounds good.
Easy stop, please.
So, folks, we're going to take a look at some imaging here, and we're on a line here that's almost on a east-west line again. Here, you see another crack leading off to the north. We're looking east. See bits of this white material breaking off and floating away. Want to get an idea what this material might be.
We have interest in the bottom right, inside that cell, maybe?
Yeah, no, the interest was up here on the right-hand side. There's snails up there where the white's coming off, that microbial active material there. Right there. Ah.
Come back down a little bit. That little,
Looks like there's a worm polychaete up there.
yeah, a couple of them. Move down a little bit. That looks like the source.
Jeez. Yeah, it looks like it's microbial. Then this looks like it's sponge-like. But I'd go.
You see all those white smokers?
Yep.
Or the snow blowers?
Yep.
Is there a temperature probe on the vehicle?
There is. Our current temperature is 4.82, so we've actually come up a little bit. We were at 4.5, 4.7 before. Interesting how these margins on the mussels are. It's like the new periostracum being put down, or shows age or exposure.
That's a very interesting area.
Yeah, well, it certainly caught my eye with that white material and the activity there, and its covering of the shells. Gastropods are certainly abundant. Chitons too.
I'm just going to try and get it tight if I can. And pull out. Pull out more. Yeah, give me a little more perspective.
Just to the first mussel to the left of that wall of white you just zoomed in on, I see antennae coming up behind there. You see them?
Yeah, go ahead. Just don't float forward. Yeah, I'm trying to.
It's behind something anyway. There's some antennae from down below too, down right on that same wall. Interesting.
Hi, Tim. It looks like there's an antenna right by the lasers. We're just above the lasers now.
Okay. He's sitting down now, so I think we're going to go in, but that's good to know. It should be just down below us now. Okay, so maybe, was I seeing that? I don't think I was. Down below, see it? See it down below, right in the corner bottom? There's two. Of course, now I don't see it. There it is. But I can't tell if there's a shrimp attached to that antenna or not. We heard shoreside that there's also one below here too. What do you think? Wait for it. Wait for it. I don't know.
far side, but I think we're closer to this one. Looks very shrimp-like, but yeah.
Something just moved. I think it was a gastropod.
Let's go up.
All right, let's. All right, we can push on from here. Go toward EC.
Sirius should basically be sitting right on top of it.
Cover a little ground. We're going to get back to the Seep area and orient ourselves again, and we continue north from there. What is this?
The tether's a little low. I don't know if I would come down. Not sure how far your tether actually goes down there. Has our temperature changed since we left that microbial area?
4.79.
About the same.
Yep.
See that white? Oh, that's just glare. Oh, never mind. Neither am I. I might even blow it out. Come more right. Run the. If you want to run the edge of this seam.
Yeah. Right. So there's a little fracture here, like a little wall, and then all this bivalve population is piled up. Seems like against it. There's the marker. That's helping us orient a little bit. Thought it might. We're heading due east now. Depth is 1117. So now short side, you can see the bubbles just almost in the center screen now coming into view. We are, yeah, there's a rise there just to the ridge. We're going to look over there and document what we see here in the field. Central, or maybe calling the central part of the field, because to the 50 meters north of here, there was a seam of mussels. We called it mussel seam. That I feel like that's kind of where we started this active seep area. There's our marker in the field of view. So we're now looking almost to the north at those bubbles.
So we want to image this right here too. Is that what I'm hearing? Wow. All you can image.
So we're going to sit down here a little bit and see what we see here, and then we'll move over.
Just
What?
small bubble release out of this clump of mussels. We are settling down to image.
Interesting.
Yeah, oh, there's it again. You got
Yeah,
it.
so we've got a little bit of methane coming out of this area right here. The odd bubble once in a while. Again, we saw probably two very minor streams coming near that main stream that we came onto earlier. Not surprised that this area is just a little leaky.
Okay, I'm stable.
But
Let me try and go in here. It's right back in there, but I see it right behind there. You go. Yep. Can you drop this a little bit for me? Thank you. Thank you. Oh.
scene looks familiar. Seeing some shiny gastropods. Look at that guy. My vent logger is locked up again. Chitons here. A lot of little mussels. What's that? There's a fish coming in the top left. You see the fish up top left? Coming into the field of view.
Very cartoonish.
My vent logger. There's also a polychaete at the top left, top center.
Might be an eel.
That's an eel pout fish, it looks like to me. Yeah, yep. So if you want to do some zooms here, that'd be good. Yeah, get the fish, get the polychaete.
What fish first? Oh, let's get the polychaete first. Okay, right there.
So it's like there's one above it too, Pilot. You see methane escaping out just there, right in front of that polychaete. You see tube worms here.
Yeah, there's one on your left.
So what we've got here is a nice active seep with mussels that are have their siphons out. We've got an eel pout fish, and you're seeing bursts of methane coming out. We've got limpets here. There's you see the methane going again. There's a shiny snail bottom left. Over the left, this fish is approaching the ROV. We've got limpets here. You can see just to the right of the fish, there's a gastropod limpet. It's like a single shell cover over the snail. And you can see one on top of a snail here, right in the center of your image. And lots of baby mussels here, lots of small recruits. Looks like they're coming in on top of existing mussels. There it goes. He's just getting situated. Those fish, in many cases, are endemic or only found in seep-like areas like this. So those polychaetes up there are really endemic or only found in seep-like areas like this. So those polychaetes up there are really tremendous. I think that's what that is. So we're here at 1,120 meters depth. We're sitting on the seafloor. We're near an active methane release that we've seen. Bubbles coming out of the seafloor. This is a place where chemicals or nutrients, hydrogen sulfide, methane, are percolate up through the sedimented seafloor of the Gulf of Mexico, and they provide nutrients for this ecosystem. You see the methane coming up. Mussels are capable of using methane and hydrogen sulfide as nutrient sources to be sort of the foundation species for these kinds of communities. Seeing small mussels, seeing limpets, just like a half-shell gastropod. There's two tube worms up to the right-hand side. You can just see them in the foreground. You can see these red animals here are polychaete worms. Looks like several of them here, extending out, crawling over the mussels and snails. That's great.
Straight left. Yeah. I'm curious. There was a little bit of
Something like that.
over there. I think it was just more polychaetes, but I'm not sure.
It's really interesting to see the pulse of the methane here too. That these aren't always constant streams that are coming out. There can be these steady bursts. There can be, I think, bursts that really come and go with longer intervals. To the bottom right here, you can see lots of little mussels that are coming in to colonize. And you see chitons here too. Right of center. I don't see it. Right of center. Those are yeah, those are byssus threads to me. Those are where the mussel is contacting with another mussel, and it can pull itself along with those. Like I said earlier, it's kind of like Spider-Man shooting out a web, and then it can pull itself closer to that mussel above it, and it can also break those threads if it wants to. Pretty neat. And so you see the larger white things on the shell, right? So it'll break those, and then you'll end up just seeing those larger white things. And that's where mussels were once connected, right? So they'll break those strands right there, and you'll see the remnants, which is just the white part, a little bit of thread. There's an ophiuroid.
Hi, Tim. This is Hui.
Yeah. right below the threads we're looking at. There's an ophiuroid coming out from underneath the muscles. Yeah, go ahead, Hui.
Yeah, so that's what I was going to ask about. Bob Carney said he saw an ophiuroid, and I haven't spotted it yet.
Roger that. I can't tell if this fish is trying to eat something. There's an ophiuroid right there coming out from below the viscous threads. Yeah, I just see 2 arms, maybe 3, moving. But love it if he'd just come out, and we could see who that is. Trying to see what else I see. You can see there's a lot of habitat here in the interstitial parts between the muscles. You can see on the edge of the muscle here in the bottom right corner, there's little baby mussels, and I think maybe also amphipods. Muscles just all clamped down. Could be because the fish is right on them. So the egg cases are on the snails here. If you look center screen on that guy, those things right there. There we go. Not sure I can see it from the back row. I'm assuming that's what that is. Earlier I thought I called them amphipods. I thought I saw some of those guys moving, but maybe they're egg cases. They're not, but you can see a difference in the composition of what's there when maybe there's less flow. We really can't see inside there. Certainly have the chitons and some gastropods here. Oh, that's great. Yeah. Okay. Seems to be a ridge up here with. Just curious if we sit in these live areas, if we're going to keep seeing methane coming up. Here we go on a little. It's like a push-up of carbonate here. Coming down on some valves piled up. What's this red thing, right? Is that a red thing right there? No, it's just a part of the shell, yeah. Right. A little bit of a wall here. Looks like lab mussels. It's a good survey. Some snails on the rock. What is that thing? See the open space between the 2 mussel groups? There's to the left. There's gastropods and something else white, like a brittle star. Do you see it? Left side of the screen. Sorry, 1/3 the way from left to right. Okay, edge of screen. That's a crab. Up right there. Up, right. What is that? Oh, it's a rock. Give me a break, Shank. All right, I'm done.
It's like a fossilized shell.
Yes, it's pitted. It's old. Yeah. Just wishing for a Galathea crab, I guess. You want to pivot more right to center up the bubbles, or what do we think? There's some. Oh, roger that. There's bubbles all over the place here.
Yeah, there's a whole string of them. But just hold there. I'm just going to mess with the iris and see how well they look at different exposures.
Living amongst these shells right now while we're looking.
I see a snail on a couple snails
On the left, yeah.
There's a live straight ahead. There's a live bivalve center screen bottom.
Oh yeah, and there's a shell centered right on the laser too, right between the lasers.
So there is some live stuff here, but certainly not much. Although there does seem to be a muscle patch just below the camera. I'm going to check down a little bit. Yeah, go right ahead. Couple live. Yeah, couple live.
What happened?
Those shells are just so bright that I've got to turn my iris down. Trying to bring it back up there. Should go to there, but I'm going to touch it.
All right, so.
So we've got bubbles here, but not the kind of lights that we had at that other place.
No, so you see, folks back home, Tom and all, that we do see little bubbles here coming out. Methane, sort of in a line right here. Yeah, we should probably get some focus on the bubbles. It seemed like they were the ones at the crab were a little more. Is that a tube worm on the right there, right of the screen, coming up?
Definitely possible.
Yeah, I think it's all.
Far field, just the right of that one too.
So maybe. Okay.
Somebody's home. Yep.
Nice. Nice.
And focal distance equals the amount the vehicle's popping around. So. Alright, get the guy still on it.
Thank you, pilot. I'm thinking about trying to find the live areas here to characterize the animals that are here and look for the methane. And there's two places. There's a mussel seam right at 16-7, and there's a 16-9, which is a nice lake of mussels. Look at this. There's bubbles everywhere. Look at that.
There's some microbial matting or something down there. Bottom left.
Sorry, Nav. Okay. Yeah, you see. You know what? You see the brine staining underneath there too, right? You see that white and dark staining. It's probably briny, but we can see if there's any animals associated with this. Look, it's pretty extensive here. So Nav, sorry, we got those two spots. I think we're going to want to keep heading then to the mussel seam. I think you're right. Yeah. Roger that. Roger that. Yeah, anemones here. You see these? Is that an amphipod on the shell there too? Looks like there is. Okay, I'm looking just south of the midline. No, that's a chitin up top. See the chitin up top? Yeah, just straight down. 6 o'clock from there on the broken shell that's sticking up. Okay, go, stop on the edge. Left edge. Huh? 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 Yeah. It's on the bottom edge. It's on the bottom edge. Top edge now. Center. Dead center. I think it is. Maybe it's not. Maybe it's not. No. Now it's on the right side of the screen. It's just at the crook. It's just at the crook. I think it. Never mind. It's fine. Straight down. It hasn't moved for a while. I thought it was moving. Is that not it? No, no. I had it right here. I should just look down. Bob Carney can tell me where everything is. Thanks, Bob.
Just to the 8 o'clock or so of the humidors and otherwise red creatures, but I'm not sure what it is.
Yeah, isopod.
Is
I think it's an isopod. Are those copepods swimming over there to the left?
Yeah.
See them?
Get the isopod. Full zoom.
Tiny. Is there any place where you can see the methane actually coming out of the seafloor? I think it's all behind rocks. I mean, shells and whatnot. Right. There's another amphipod. Seems like we just had this open area of brininess. It could be a line of this discoloration. Right. There's another amphipod. Seems like we just had this open area of brininess. It could be a line of this discoloration.
That's where we started, I think. 3 meters? I don't know if we've been through. That might be our real, the big bubbles.
So now, did you see these on the sonar, these bubbles? Did we think this is what we were looking at, guys?
We
Definitely saw these? Nice. Okay. I remember seeing it. I just wasn't sure if we felt this is where we are. Roger. Okay. Wow, that's pretty awesome. Able to see those. It was clear in the sonar. There wasn't really much doubt about it. Okay. If everybody has seen enough of this, coming out. I'm going to come to the right. Yeah, there you go. That's it. The stream right there in the center left seemed more vigorous, and I think people were saying let's center up on that. Huh?
When we came over here, Dave, there was two, in Little Herc sonar, there were two distinct. What's the lifespan of a muscle and how long do the shells last?
Tim is actually out. It's me again. Sorry. It just changed.
I'm just curious about the ratio of dead to live. So what's the lifespan of a muscle and how long do the shells last?
That's a very good question. I'm gonna have to relay that to Shure. So we were wondering what the average lifespan of one of these mussels are. I mean, we're seeing a lot of empty shells here. So you know, we have a lot of empty shells relative to the live mussels we've been seeing. And does that say anything about the lifespan of the mussels, or do we know anything about how long these mussels live? So it looks like we've been chasing bubbles all afternoon, and we'll continue to do so as we head roughly northwest towards the area um, where you can see these mussel seams. You know, it's like the mussels are kind of following the fault lines or cracks in the seafloor. I'm guessing that's because those are the places where the methane would bubble out. For most of the afternoon, we deployed a physical marker, I believe it is called M28, next to the first seeping 5 p.m. And there will be a time change on the ship later tonight, but I guess that doesn't matter because this is the last dive. Um, so it's gonna still be Eastern time for this dive. And we're coming upon some scattered carbonates. I'm seeing a few patches of live mussels here. A number of urchins on and close to the carbonate rocks. There's probably a crab on the left. Oh yeah, that is a crab. I see a couple of tube worm tubes.
It is.
Ah, okay, we're coming upon the mussel seams. You can see that they're arranged in a pattern that seems to follow what may be faults or cracks in the seafloor. I think there are also more tube worms here, at least a little more than the area we just passed. And they seem to occur pretty close to the mussels here.
Reginald, if you would stop us here, please.
Oh, thank you, Peter. Okay, so we do know that mussels of the same genus, which is Bathymodiolus, in hydrothermal vents are known to live for as long as 18 years. And their shell size can go upwards to 14 centimeters. So those are Bathymodiolus mussels from hydrothermal vents in a Lao basin. But I don't know if that would be the same for the Bathymodiolus mussels here. We're continuing to move around this area with the mussel seams, where the mussels seem to follow fissures on the seafloor. looking for interesting biology to image. Can we come down a little closer to the bottom? I'm not seeing any signs of potential bubbles in a sonar or in a video. Not yet.
Dave, my sense was that this seed sort of started and I was falling into it.
I may just have to move the ship to get you guys a little.
Okay, you can see a lot of those gastropods and the life muscles here. At any time, please let me know if there's anything you would like to image or look at. And of course, at any time, if you want to have a certain view on the one of the feeds, also just let me know.
Text you just do some macro.
Okay. Yeah. Why don't we look at look at closely one of the some of the muscles along this seam, right? Maybe with the crab or the urchin in the background. Would that be possible? If we sit here, do you think we would be able to see those tube worms on the right now? Okay, that sounds good.
Pilot, you are very, very soft. Would you move your microphone a little bit closer?
Sure. How's that?
Thank you. Hey, there's a sea star. Haven't seen one of those yet. Left field, center.
You see a sea star?
There was.
So we have a potential sea star here. Yeah, let's start with looking at the tube worms, please. Thank you. Okay, looks like the tubes are about 20 centimeters long.
Okay, we're pushing it on video. Go ahead. Pushing it on the head. Just check.
Close-ups of the tube worms. I'm still guessing that they could be lamellobrachia, but I'm not really sure. In addition to their symbionts, a lot of these tube worms are also known to use their roots in the sediment to mine for sulfides.
So far we're pushing all the way towards it.
Okay. Close up of the tube worm. Okay. And yeah, after we do a close up of this, can we sell it too?
There it is.
Yep.
Just came out of screen.
Yep.
Is it tight?
On the tube.
Oh yeah, there it is.
And we're pushing the rest of the way. Yeah, that's all I got.
So it's on the tube. Box right now. See if there's anything on it. Oh, you do see those tiny tube-like structures, right? Oh, and then there's something right in between those rocks. Okay. Okay. Okay, let's us and then the urchin just below them. Oh, I see the sea star, bottom left. Okay, yeah, let's add the sea star to the list too, if you could. I'm on the crab. So we see this crab, sea urchins, tube worms, a sea star here. Yeah, you can see the encrusting barnacles on the crab. That's a lot of detail right there. Ooh, look at that. That looks very ticklish.
You can see Featherheart's lights in the crab's eye. Oh, look at that.
Yeah. Oh, yeah, look at that. You can see the two lights of the ROV reflected in the eye of that crab. If only we could zoom in more to that eye, maybe we can, clearly see the ROV in it.
That would be a funky shot. Yeah, focusing on the mouth of the crab.
Okay, we're gonna take a quick look at the mouth now. Okay, yeah, let's come. Okay, zoom on urchin. You can see a lot of detail here on the urchin.
That looks like that would hurt. Alright, I'm gonna back out. That should be good detail.
Cool.
For a while.
Okay.
How close in do we want to go?
Let's I wanna. Ooh, okay. Okay, so you see how on the top left, some of the gastropods, their shells are a little more shiny.
Yeah.
Yeah, can I look at that?
Sure.
Thank you.
I'm gonna push in on the shiny gastropod. Shiny gastropod up.
I guess maybe for the other gastropods, their shiny shells have just been covered by, that kind of white, dusty looking material. Maybe it's the same shell, but I can't tell.
Focus on the opening of the tongue.
Okay. I think that's the siphon of the muscle that you're seeing on the right. Okay. So that's a nice close-up of the gastropod. Okay.
Really nice shot. Real action shot from the crab there. Okay. So,
Yeah, I believe they're to the left now, right? Oh. Okay. That's still a really good shot. Ooh. Ah. That's a great shot of the squat lobster. Look at its furry arms. Oh, and it's moving, I believe, some of its mouth parts. I always feel like I'm being tickled when they do that. Okay. That's a great profile of the squat lobster. Oh yeah, yeah. What do you see? Oh, the white stuff. Oh, okay. Yeah, the white spongy stuff. So what is that again? Okay, is this a max zoom? Okay. Identified this. Just to the bottom right. Is that what you're saying? Oh yeah. Possible baby muscles. Bottom right. Oh, top right now and right now. Interesting. Okay. Zoom on starfish. We're doing a close-up of a starfish that's partially on top of a mussel shell. The other half is on sediment. I don't see any obvious activity, but we're zooming in even closer now. Interesting. You can see a lot of detail of the texture on this starfish. To Tim? Okay. There's a little bit of sedimentation on this sea star. The edges look a little furry. Okay. We go chase more bubbles after the crab. Ooh, look at that. There are two crabs on top of each other. Well, one on top of another. I don't think. Yeah, I think we did. Yeah. I mean, I'm guessing this is some sort of mating ritual. You know, it looks like the crab on the bottom, like, you know, all of its legs, right, are folded up and retracted, kind of. Yeah. Let's move the light away. Okay. Um. Well, I mean, I'm kind of interested in looking for more bubbles, but I don't know if there are any here.
No, not really. From the earlier transit, these muscle seams just kind of ran all over the place. So I think you can just kind of follow them and go off on different branches. See if there's anything different.
Another urchin. And it looks like, those gastropods are mostly on the muscles that are along the seam.
This is video. Did you get those figured out?
Our current depth is 1119.8 meters. We're 0.4 meters above the seafloor. Water temperature is still 4.7 degrees Celsius.
10,
Yeah.
9 degrees. That clump.
Oh, yeah, that looks like a pretty
Might be interesting to
intense
see
clump.
bubbles.
Yeah, they look like they're pretty tightly packed. This tightly packed group of muscles are among a lot of empty muscle shells. It's kind of like in the center of a field of empty shells.
Look at this clump a little bit, see if there's any sign of. Sure, third is in, bubbles, go for it.
Quick zoom on the muscles. Okay.
On the left. Snails.
You can see that siphon right there coming out of that muscle.
Maybe another one on the lower right. Tube in the distance on the top left, the very tip of it sticking out. More snails top right.
We're seeing, your usual gastropods and chitons on these muscles.
Yeah, I don't see anything but those. Get a little more. Give me a little tilt up, please. That's great. I haven't seen any hint of a bubble since we came over this direction.
Yeah. Even though the muscles are tightly organized around these seams, we're not seeing any bubbles coming out of those seams.
No, that one right there, that one's got occupant. Might have somebody home.
Yeah, it looks occupied.
That's almost it for signs of life in this neighborhood right there. Just a little polychaete on the tube.
Yeah, and I'm guessing this is also a lamellobrachia, maybe? I am not getting a lot of response from Shoreside right now, but I'll ask again. So the idea right now is head about 40 meters to the south, and then head west from there. That's an area we haven't explored much during this dive yet. That idea sounds good to me. If anyone I'm sure has any other ideas, please let me know. In the meantime, let's prepare to go in that direction.
New direction travel is going to be 170. Copy that? Looks like there might be a lot further ahead plan.
We're now moving south. Yeah, this is another tightly packed group of mussels with 2 urchins on it. Yeah, no bubbles as of yet. Some carbonates in the background.
Bridge now. My goodness. I'd like to go 50 meters, 170 degrees, 0.2 knots, please. Thank you.
And the ship is moving. We're seeing a very dense, high concentration of shells here.
One with the vehicle.
Even though we've come across several, tight packages of mussels, we haven't seen any of them, emit any bubbles. Oh, that's a crab with its arms held high.
Mission for bubbles here, is that? Yeah, I think we're going exploring.
Yeah, exploring while keeping eyes peeled for bubbles. Oh, fish! Fish? Yes, fish. Fish.
Mussel shells if we have time to work our way that far.
Many scattered shell fragments here. Another tight clump of mussels. Another mussel, same, I believe.
Yeah, I think so.
Crab on top of shells. Our current heading is 186, depth 1119.6 meters, 0.6 meters above the seafloor. We're going south and possibly west after that to explore the unexplored regions of this mussel field. Maybe we'll even find the west edge of it. And some carbonates on sediment here. These carbonates are not covered by mussels, but oh, okay, there's a crack in the middle, which is another mussel seam. Perhaps when the crack first formed, this was a pretty actively bubbling area. But maybe that was a long time ago, so it's not very active anymore, but I don't know. A lot of empty shells here on the sediment. Oh, what's that swimming in the water column just? As we explore this really, really densely packed region filled with empty shells. Okay. Okay.
I believe the time is to run west once the ship stops its move. It should be very soon.
Another fish.
Yep.
Fish. Crab.
Some white staining.
Yeah.
Not much in the way of large shells though. There's a couple right there, but yeah, not as many.
Huh. Well,
That in the water column.
oh yeah, who is that? You know, I think the shell suddenly became a lot more dense here, did it?
Yeah, I agree.
What does that mean? You know, investigated pretty extensively.
No, we saw it and continued past it.
Ah, okay. Well, let's see. Well, let's see what the group would like to do. So we could move west to kind of complete, right, the third side of this triangle. Now, there aren't any targets that say possible seep in that direction, but, you know, I believe we discovered bubbles today that weren't very close to those possible. Currently moving over a region of lots of empty shells. Our depth is 1,120.6 meters. We're heading west. To unexplored region. Right now we're just seeing a ton of empty mussel shells with no obvious staining in the sediment.
Is it hazy or is it not? It's hazy.
And apparently it's getting a little hazier.
shot 90 home.
Our heading is almost exactly due west now. What's our speed?
0.2.
0.2 knots, okay. Where can I see that?
There's no real way to see that.
There's no real way to see that? Okay. Okay. Mm-hmm.
Kind of like looking for Easter eggs.
Oh yeah. That's great.
Got a little redeemed on that one.
Ooh, okay. We have a little more dark and light color staining here. I don't see a lot of live muscles yet, but definitely a lot more staining just now.
There's some live muscle.
Ah.
What if those black long filament things right there is what I was looking for.
Oh, those things. I don't know what that is. Can we do a quick snap zoom on those long thingies? Bottom right of lasers.
See it, Jeff?
Thank you.
It is. It's
Oh,
retracting like an Akira.
oh my God! Look at that guy. Oh, oh, okay. Okay. Oh yeah. Oh wow. Look at that. So that's what a sipunculate pharynx looks like. They are indolithic.
Of course they are.
Wow. I have never seen them in action like this before. And those tubes right on top, they seem to be covered in this white kind of cottonish material. Wow. Yes? Oh, sorry. Yes, Jeff, go ahead. I'm sorry. I was just so concentrated on that pharynx. Um, what? What? Okay. Uh, okay. Zoom on tube worms. Okay. Oh, sure. Okay, so on the bottom one, right, you can see that there is, when it's focused, that is, you will see, it's just in the, oh, I guess not. It retracted. Oh. Covered by bacterial filaments. Okay. Zooming out slowly. Yeah. Really? A nuggle. So we just passed by a couple other sea urchins in this stained area. We passed by some live mussels, imaged a serpunculate pharynx, an indolithic serpunculate pharynx. And now this area is becoming a little less stained again. We pass by this area as we are exploring west. Our current depth is 1,122.8 meters. Heading 242, altitude 0.89 meters above the seafloor. We're looking for bubbles, if there are any. Yeah. Where did all the shells go? Yeah. So I guess this might be the western edge of the shelled area, but I don't know.
Yep, there you go.
Wow, look at that.
Or
Transparent
transparent tube worms. There's a barnacle on the right, looks like. The right one, and pimp.
Oh, wow.
There's a barnacle there.
Yeah, that's a great shot. Crabs. Okay. A lot less shells here on the sediment. Also, there's a apparent lack of staining, so I'm guessing this is a lot less active area. Fish on the left with 15 seconds to go. Okay.
Well, science team, it's the witching hour. It's 5 o'clock, and they're gonna pull us off the bottom here. Very highly successful dive, guys. Thanks very much. Getting excellent reports back from the beach. People are very excited. Did really good work today.
Lots of cool discoveries. Yeah, during the past 3 weeks, and for putting up with me.
Yeah, I think we should thank the EX science team for this leg, 1202. Thanks, everybody, for all your input and help.
Yeah, thank you for the amazing driving and piloting and imaging.
This mode of exploration is a new one to many scientists, but they are taking it on, and I think you're seeing the real value in this. As you can see today, tremendous results. We surveyed shipwrecks, found maybe demasting in one case, and found new seep communities, new coral communities. We've imaged more than 500 corals, maybe 600 corals. Oh, yeah. And anyway, it's been a great cruise. Thank you, guys. Appreciate it here in the room. Appreciate it back on the beach. Thanks, everybody.
Yeah, thank you very much, everyone. So I guess this concludes leg 2 of the cruise.
Yeah, there'll be a leg 3 starting on Wednesday, and they'll be exploring the deep Gulf of Mexico to the west, where we've been. They've got 18 dives on that program, and I know they're busy right now putting those targets together. They've got 18 dives on that program, and I know they're busy right now putting those targets together for us all to enjoy. And I'll be doing that back on the beach. And thank you, Penn, for all your hard work. Really appreciate it.
Oh, thanks, Tim. Yeah, so please stay tuned for leg 3.
Thank you all. We'll be in touch.
Thank you very much.
Remember, too, that all this data will be on the FTP port.
Thanks to the whole team. That was a really great dive today. It was exciting to see those bubbles. Really appreciate it.
It's our pleasure. We're all learning it together, so I think it was fantastic. All this